In the arid, sun-scorched cliffs overlooking the Dead Sea, a secret lay concealed for 2 millennia. In 1947, a young Bedouin shepherd was searching for a lost goat when he tossed a stone into a cave and heard the unexpected sound of shattering pottery. What he and his companions uncovered in that cave, and 10 others nearby, would become known as the greatest archaeological discovery of the 20th century, the Dead Sea Scrolls. These were a library of 1,000 ancient manuscripts, the oldest surviving copies of the Hebrew Bible, and a collection of previously unknown religious, legal, and philosophical texts. Written between the 3rd century BC and the 1st century AD, they opened a window into a pivotal moment in human history, the era that witnessed the evolution of modern Judaism and the birth of Christianity. But this discovery was also a colossal puzzle. The scrolls had disintegrated over the centuries into more than 25,000 fragments, some, larger than a postage stamp. Scholars have long debated their origins. Were these scrolls written by a small group at Qumran, or were they the works of various groups in ancient Judea? Then scientists extracted DNA from the animal skins the scrolls were written on, and what they discovered proved something shocking. Some scrolls didn't come from Qumran at all. Found between 1947 and the 1960s in the 11 Qumran caves and other desert sites like Masada, the Dead Sea Scrolls are written mostly in Hebrew, with some in Aramaic and Greek. They come from a time when Greek culture influenced Jewish life, sparking groups like the Essenes, Pharisees, and Sadducees. Sorting these tiny fragments, some as small as a coin, is like solving a puzzle with missing pieces. Scholars use handwriting styles and text content to connect them, but questions remained about where they were written and by whom. Until now. To understand this breakthrough, we must first understand what the scrolls are made of. The vast majority were written on parchment, a durable material made from processed animal skin. For molecular biologists, this was the key. Every fragment of skin, no matter how small, contains a trace of the animal it came from, its DNA. But extracting DNA from 2,000-year-old scrolls is extraordinarily challenging. Ancient DNA is fragile, broken into small pieces by time and heat, and contaminated with modern human DNA from people handling the scrolls. The team studied 39 items, including 26 scroll fragments, plus sandals, water skins, and a garment from desert sites. To avoid harming these treasures, conservators collected tiny samples, sometimes just dust, from blank areas of the scrolls. The team worked in an ultra-clean laboratory in Sweden with airlocks and UV lights to avoid contamination. They used a method called silica-based extraction, which captures DNA molecules. The first step was determining which animals the scrolls came from. The team compared scroll DNA to animals common in the ancient Near East, like sheep, cows, and goats, matching it to known DNA patterns. Of 26 scroll fragments, 24 were sheepskin, but 2, 4Q70 and 4Q72b, both from the book of Jeremiah, were cowhide. This might not sound dramatic, but in the context of the Judean desert, it was a revelation. The arid, parched landscape around Qumran is suitable for raising sheep and goats, but not cows, which require substantially more water and pasture land. There's no archaeological evidence that cows were ever raised or their skins processed at Qumran. The conclusion was inescapable. These scrolls were likely made elsewhere, probably the Jordan Valley, and brought to Qumran. The Jordan Valley had long been known for its fertile land, ideal for agriculture and livestock, including cattle. This finding shatters the old assumption that the Qumran community was a completely isolated sect, writing all its own texts. Instead, it suggests that Qumran was a vibrant intellectual center, more of a library where scrolls from diverse origins and different textual traditions were collected and studied. With the animal species identified, the researchers turned to sorting fragments, using the insight that fragments from the same animal are likely from the same scroll. As ancient scribes typically used one animal's skin per manuscript, they analyzed both mitochondrial DNA, inherited from the mother only, and nuclear DNA, inherited from both parents. Sheep have 5 mtDNA haplogroups, which are genetic lineages passed through maternal lines, labeled A, B, C, D, and E, with A, B, and C common in the Near East. The team's primary focus was a non-biblical text, a mystical composition known as the Songs of the Sabbath Sacrifice. Copies of this important liturgical work were found in the Qumran caves, suggesting it was significant to the community there. But here's the twist. Another copy of the same text was discovered over 50 kilometers south at the famous mountain fortress of Masada and is known as Mas 1K. This created a debate. Did the text originate with the Qumran sect, and did some of its members later travel to Masada? Or was this work popular among different Jewish groups across the land? Once again, DNA provided the answer. The researchers found that the multiple copies of the songs from the Qumran caves were all made from the skins of genetically related sheep, as they all belong to haplogroup B. This strongly suggests they were all produced locally from the same flock by the same community of scribes. The Masada fragment, the Mas 1K, however, told a different story. Its DNA came from a genetically distinct line of sheep, which belonged to haplogroup A, not related to the Qumran flock, which were all haplogroup B. What this means is that the community at Masada didn't receive a scroll from Qumran. They made their own copy of the text. The Songs of the Sabbath Sacrifice was therefore not the exclusive property of one sect. The songs were popular beyond Qumran, circulating among different communities in Judea. The final challenge was one of provenance. Because many fragments were not excavated by archaeologists, but bought from antiquities dealers in the mid-20th century, their true origin is often uncertain. Some fragments were assigned to the Qumran caves simply because they were part of a collection being sold at the time. One such fragment, 4Q344, has always been an oddity. While nearly all other Qumran scrolls are religious literature, this piece is a legal document, a note acknowledging a financial debt. Its script and later dating also set it apart. Scholars have long suspected it didn't really come from the Qumran caves. The genetic analysis confirmed their suspicions. The sheepskin DNA from this financial document did not cluster with the DNA from the main Qumran scrolls. It was a genetic outlier. This provides the first piece of hard physical evidence that this fragment was likely found at another site in the Judean desert, where such everyday documents were common, and was later mistakenly mixed in with the Qumran collection. Similarly, a fragment from the book of Isaiah was found to be a genetic outlier, suggesting it too originated from a different, as yet unidentified location. These findings show that some fragments were misattributed to Qumran due to their acquisition through antiquities dealers rather than controlled excavations. This genetic detective work has profound implications. For the first time, theories about the origins and relationships of the scrolls have been supported by independent scientific evidence. The genetic data has acted as a powerful arbiter in decades-old debates. It shows that the Dead Sea Scrolls are not just the product of a single isolated group, but the results of the work of various groups that shared the diverse cultural and religious landscape of Judea during the Hellenistic Roman period. Some scrolls were likely written by local scribes at Qumran, adhering to their unique scribal traditions, while others were brought from distant places, carrying with them different versions of texts and ideas. This diversity mirrors the genetic diversity of the animals whose skins were used, and as research continues, new discoveries may yet emerge, revealing further connections between the scrolls, the people who wrote them, and the broader world of the ancient Near East. Thanks for watching. If you enjoyed this deep dive into how genetics is rewriting history, make sure to like and subscribe so you don't miss the next discovery. This is Hidden Strata.